my talk is on freeze tolerance plasticity in the intertidal mussel Mytilus trossulus. So this talk is based on a paper. It's currently under review at the Journal of Experimental Biology with me and my two co-authors. So our study organism is the bay mussel, which is an intertidal mussel found all up and down the west coast of North America. And the reason why Mytilus trossulus needs to be freeze tolerant is because they're sessile species. And this means that when the tide goes out in the wintertime and temperatures drop below zero, they're susceptible to sub-zero body temperatures and therefore freezing. And freeze tolerance is a very unique tolerance that not many organisms have. So we wanted to dig more into it and understand some plasticity and some potential mechanisms behind that plasticity of freeze tolerance. So what did we do? We collected mussels from Tower Beach, which is in Vancouver, and due to its proximity to the Fraser River, this is an estuarine environment. And then we brought the mussels back to the lab, experimentally exposed them to one out of a variety of sub-zero temperatures, and assessed their survival. So we wanted to determine how plastic mussel freeze tolerance is. So we tested how the extent of mussel freeze tolerance changes with the following three factors. So our first prediction is in terms of seasonality. We would predict that mussel freeze tolerance would increase in the winter months, which is intuitive. Next, in terms of intertidal shore height, we would predict that freeze tolerance would increase with increasing shore height. This is because mussels that are located higher on the shore are exposed to air for longer amounts of time on a daily basis, and therefore they're exposed to longer amounts of cold and potential freezing stress. So those high intertidal mussels might have increased freeze tolerance for that reason. So what did we find? Basically, we found that support for both of these two predictions. So on the y-axis here, we have lower lethal temperature. So that's the temperature that causes 50% of the mussel population to die. So with a decrease in lower lethal temperature, that's actually an increase in freeze tolerance because the mussels are surviving at lower temperatures. So in terms of seasonality, you can see as we go from July to November and January, we're getting a nice trend of a decreasing lower lethal temperature, which supports our prediction. And also with shore height, we're seeing that high intertidal mussels are in fact more freeze tolerant, particularly so in November and January, again because of that lower lethal temperature. So our third prediction is that high salinity acclimation would increase freeze tolerance. So why might we predict this? Well, freezing is actually an osmotic stress. So what this diagram is showing is as freezing is occurring, ice formation is happening in extracellular spaces. And this is actually concentrating the extracellular fluid and creating an osmotic gradient. But mussels are actually really good at tolerating osmotic stress. And how they do this, they're osmoconformers. So they will accumulate intracellular pools of osmolytes in order to balance their intracellular osmolarity with the extracellular environment. So these osmolytes might be helping them to cope with freezing by helping them to deal with the osmotic stress associated with freezing. So what were our results? Well, we found support for our prediction that high salinity acclimation does increase freeze tolerance. So again, we have lower lethal temperature on the y-axis here, and we compare between 15 parts per thousand salinity, which is brackish salinity, and 30 parts per thousand, which is more so similar to ocean water typical salinity. And we're seeing a really strong decrease in lower lethal temperature, therefore an increase in freeze tolerance in those high salinity conditions. And the magnitude of this change is quite large as well, which is important to note. So what we wanted to do is determine what the mechanism might be behind these changes. So we measured how osmolyte concentrations changed in all of the factors that we tested. So these are the 5 main osmolytes we were looking at. In terms of season, we saw an increase in these 5 osmolytes in the concentration when we compare between August and November mussels. In terms of intertidal shore height, we saw no change when we compared between shore heights. And in terms of salinity, we saw an increase in the osmolyte concentrations in higher salinity. So this means that osmolyte accumulation correlates with increased freeze tolerance both seasonally and through high salinity acclimation. So osmolytes may be a potential mechanism underlying this freeze tolerance plasticity.